All right, our second half of chapter 1, which is what is in a human genome, is going to include three learning objectives. We're still going to be differentiating between some of the different types of genetics vocabulary that you'll be using throughout the entire year. We're going to continue on and identify the various levels of genetics. We started those first few levels in the last PowerPoint. We're going to finish them up in this one. And then we're going to start seeing how can we apply genetics to the real world. What are some examples of that? The next level of genetics is the actual human genome. Remember, your genome is your entire collection of all your DNA that's found in your cells, all of your genetic information. And so our body contains approximately 37 trillion cells, and all of those cells, except your red blood cells, contain your entire genome. Now, your red blood cells don't contain your entire genome because red blood cells, as they mature, lose their nucleus. Well, most of your genetic material, of course, is in your chromosomes, which are found inside your nucleus. But we also have mitochondrial DNA that we can't forget about as well. And so we'll talk more about that in a later chapter. But most of your genome is found in the nucleus within your chromosomes. Now, the expression of different genes to manufacture proteins is what drives cell differentiation. When you think about it, all those different cells in your body all have the same DNA. They all have the same genes in the DNA. So why is it that a skin cell functions as a skin cell and a liver cell functions as a liver cell and so on? It's because different genes are being turned on and turning off that are forming some of those different proteins. And that's what we call cell differentiation. And that just means that they're becoming more specialized into distinctive cell types. Now, your stem cells are your base cells. They're less specialized, and they provide you with a constant supply of cells. And what happens to them is that they can either they can divide and make more stem cells, or they differentiate. Oops, marking up the wrong stuff here. So they can either divide and make more stem cells, or they can become specialized and help to undergo cell differentiation so that they can become skin cells or heart cells or lung cells. Some other pairs of genetics terms that you're going to need to know the difference between are genotype and phenotype. Genotype is your allele combination. Remember, we said alleles were alternate forms of the same gene. So here is one allele, the capital T, and here's another allele, the lowercase t. Well, when you always inherit one set of alleles from your mom and one set of alleles from your dad on their chromosomes that would be in the egg and the sperm cells. So together, that makes up a genotype. So you could inherit both dominant alleles from your parents. You could inherit a dominant allele from one and a recessive allele from another, or maybe you're going to inherit both of the recessive alleles. So each one of these is a genotype, your allele combination. Now, your phenotype is the physical expression of that genotype, or what it looks like, the visible trait. So if you just think of phenotype and physical expression of that genotype, that tells you what the visible trait is going to be. Are you going to be tall or are you going to be short? Okay, so make sure you know the difference between those, because we'll be using those terms quite a bit. Dominant and recessive refer to what type of allele that you inherited. Now, dominant alleles, they can be expressed only if you just have one copy. So if you're big T, little t, and big T stands for tall, little t stands for short, this dominant allele right here would cover up this one, and therefore that individual would be tall. And so all you need is one copy for it to be expressed. Recessive alleles, you have to have both of them. They have to be present on both chromosomes for it to be expressed. In other words, little t, that's the only way. So you can tell if somebody has a recessive trait, we would be able to know what their genotype is. Now, you wouldn't be able to tell somebody's genotype if they had a dominant trait because they could be big T, or they could be big T, big T. So these are some more terms that you'll need to know throughout the course of genetics. From there, we move on to family. Well, one thing we can do is we can look at what's called a pedigree chart, and pedigrees show how traits are inherited or passed on through generations in a family. Okay, through generations in a family. And so here it's showing you, here's one generation, the second generation, and the third generation. Okay, showing that trait that's being passed on. So this is showing, the one over here is showing albinism, individuals who are albino, versus over here it's showing polydactyly on the right-hand side. So you can use those charts to show how traits get passed on. And a genetic counselor may look at that and analyze those charts for a family. The next level moving up, getting broader and broader now, is population. Now, when we talk in science about a population, it refers to a group of individuals of the same species that have healthy offspring together. And then from there, we can talk about gene pool. That's the sum of all of the different alleles that would be found in a population. And so when we look at population genetics later on in the year, we look at all the alleles in a population and we can start to see that, well, some populations of individuals, certain traits are found in a lot higher proportion than in other groups of individuals, other populations. For example, cystic fibrosis is higher in Caucasians, whereas something like sickle cell anemia is more common in African American populations. So we'll talk more about that when we get to population genetics. Evolution is, when we look at that in terms of genetics, we're looking at changing allele frequencies in populations over time. So how do these different allele frequencies change within a population over time? And then we can start doing genome comparisons among species to show evolutionary relationships among them. So now that we're done with the different levels of genetics, how can we apply it? Why is it important to study genetics? What are some areas in the real world that we can apply this study of genetics? Well, one of the things is to establish identity. We can use genetics. And you may have heard the term DNA profiling. It used to be called DNA fingerprinting. And what it does is it compares DNA sequences among individuals, and it can help establish or rule out identity, relationships, and ancestry. So in cases of, if there's victims of natural disasters where all they do is have fragments of bodies, or, they have to try to identify who this individual was, and there's not a whole body there, unfortunately, if they can get some usable DNA from what remains they have, then they can match that up with maybe a family member to determine the individual. Locating blood relatives of adopted individuals, historical details of showing relationships between individuals, forensics. You've probably watched lots of different crime shows or where they do forensic identification of suspects or victims. And oftentimes, if there's blood or other kind of evidence, if it's a rape, maybe semen or something like that, anything that they can get some usable DNA from to test, they can then use that to identify possible suspects or individuals who may have committed the crime. Evidence for past epidemics. They can even look in old tombs to try to determine, what disease did that individual die from. If some of those mummies that were pretty well preserved, if they can go in and get some usable DNA from that individual, they can find out a little bit about maybe why that individual died. In healthcare, when we look at disease, a lot of times it's an interaction of genes and the environment, and so understanding that interaction between genes and the environment can be important in maybe coming up with different types of drugs or other treatments for individuals. Just the genes that you inherit can affect immunity and disease susceptibility. You think of some people who hardly ever get sick. Well, is it something in their genes, their genetic makeup, that is allowing them to be a little bit more resistant to certain diseases than other people are? Pharmacogenomics, this term right here, is a field that identifies individual drug reactions based on genetics. Stanford has a whole kind of area within their research study called Imagenetics, where they're trying to, if they could do a kind of a DNA fingerprint on you to kind of see how you might respond to different drugs. And that's what, when you talk about personalized medicine, that's about as personal as it gets. If they can match up what type of drug best suits your DNA profile, how much of the drug, can we give you certain ones so you can avoid getting, side effects, those types of things. Another thing they can look at is exome sequencing, looking at mutations in protein encoding parts of the genome to see how that may affect an individual. And here's a little assignment that you guys can do, going to Schoology, where I read about how exome sequencing saved a boy's life on page 10. When we look at inherited illnesses caused by a single gene, that's going to be different from just, other types of just communicable diseases that you might pick up. You can actually predict how they're going to be passed on. And you might be able to do some genetic testing ahead of time to see if the individual has that particular allele that would cause them to have that particular genetic disorder. And so it's a little bit different than just communicable diseases. When we look at the diseasome, this is just kind of a chart where they can kind of show how different diseases might be connected that share genes that have altered expressions. And so you can kind of see that certain disorders may also have other genes that are very closely related to those that might have some similar expression of those genes. And then when we look at global perspectives, metagenomics is sequencing all of the DNA in a habitat. Well, why would you want to know that? Maybe that's going to help understand the biodiversity of that particular area. Maybe it's going to help us find new drugs in that region that might help cure certain diseases. So it also can be used to study the human microbiome. You have more microbes in and on your body than you have of your own cells. So kind of understanding their genetic makeup too, and how that may interact with either helping your body or sometimes harming your body would be important as well. And then other things to think about with genetics and studying genomes is some of the social issues that can arise. Like, does everybody have equal access to genetic tests and treatments? What if somebody misuses your genetic information? What if somebody starts to use some of the genetic technology to harm people? And so even though we may try to improve the quality of life by using certain genetic tests, we also have to be careful of any of the